The recognition of pathogen-associated molecule patterns, PAMPs, or damage-associated molecule patterns, DAMPs, triggers a broad range of responses in innate immune cells. These responses not only facilitate the elimination of pathogens or damaged cells, but also help establish the conditions necessary for an adaptive immune response if needed. In this video, we will explore the mechanisms that allow immune cells to migrate from the bloodstream into tissues, a process known as extravasation. This occurs as a part of the vascular response, namely a change in characteristics of blood vessels that facilitate the exit of immune cells. Before diving into the stages of extravasation, let's first look at adhesion molecules, a series of glycoproteins responsible for mediating cell-to-cell interactions. From a structural standpoint, adhesion molecules fall into three main families: selectins, certain proteins belonging to the immunoglobulin superfamily, and integrins. Selectins are carbohydrate-binding membrane glycoproteins. They are expressed by endothelial cells activated during the vascular response and play a key role in initiating contact between leukocytes and the endothelium. B-selectin and E-selectin are particularly important in innate immunity. More specifically, endothelial cells rapidly express P-selectin, which is performed in viable Pallade bodies in response to various stimuli. Meanwhile, E-selectin mRNA is transcribed and expressed on the endothelial surface within a few hours. Both P and E-selectin bind to sulfated sialyl Lewis X, an oligosaccharide found on surface glycoproteins of circulating leukocytes, mainly neutrophils. Mutations in enzymes involved in synthesizing sulfated sialyl Lewis X, such as fucosyltransferase, can result in leukocyte adhesion deficiency type 2, LAD2. Proteins of the immunoglobulin superfamily can contribute to adhesion known as intracellular adhesion molecules, ICAMs. These are expressed by endothelial cells as well as by activated leukocytes and impose a stronger adhesion. The main ICAMs are ICAM1, ICAM2, VCAM1, vascular cell adhesion protein 1, and PCAM, platelet endothelial cell adhesion molecule. Integrins, on the other hand, serve as ligands for ICAMs. These are proteins made up of alpha and beta subunits, with several variants of each existing. The beta subunit is shared across multiple subtypes, while the alpha subunit differs. Key integrins involved in innate immunity responses are LFA1, leukocyte functional antigen 1, CR3 complement receptor 3, also known as MAC1, CR4 complement receptor 4, VLA5, very late antigen 4. CR3 and CR4 recognize IC3b and act as complement receptors, a topic we discussed more in detail in another video. But CR3, like LFA1, can also bind to ICAM1 and ICAM2, imposing a stronger adhesion compared to the selectins alone. Additionally, CR3 can bind to fibrinogen and coagulation factor X, bridging the immune system and the coagulation system. which is also talked about in a previous video. Lastly, VLA5 can bind to the fibronectin of the extracellular matrix, mediating the interaction between the cells and the surrounding connective tissue. Integrins are also expressed at low levels by the normal epithelium, not activated by the inflammatory stimuli, allowing for a normal outflow of circulating leukocytes into tissues. Inflammation, however, leads to high levels of ICAM1 expression by the endothelium and alters the conformation of integrins expressed by leukocytes. From a biochemical perspective, integrins like LFA1 and CR3 exist in two states: a high affinity state for ICAMs and a low affinity state. At rest, they bind weakly to ICAMs. Still, some chemokines produced in an inflammatory environment lead to the cytoskeletal protein taurine and the intracellular tail of the beta subunit of LFA1 and CR3 to bind. This bond forces integrins to adopt a high affinity conformation for ICAM1, imposing a stronger adhesion. Defects at various levels of integrin or other proteins involved in the adhesion process lead to leukocyte adhesion deficiency 1, LAD1, which causes recurrent bacterial infections and delayed wound healing. Lastly, integrins are quite useful from an immunologic perspective, since they serve as lineage markers for some immune cells. CR4 is widely expressed by conventional dendritic cells, or CDCs, whereas CR3 acts a marker for monocytes, macrophages, and myeloid cells in general. Unlike CDCs, plasmacytoid dendritic cells, or PDCs, express low levels of CD11b, CD18, but can be identified by the expression of either BDCA2, blood dendritic cell antigen 2, in humans, or BST2, bone marrow stromal antigen, in mice. Having introduced the molecular components involved in the process, we will now delve deeper into the dynamics of extravasation. Under normal conditions, leukocytes travel through the central part of blood vessels where blood flow is faster. However, vasodilation due to the inflammatory stimuli causes the blood flow to slow down and allows leukocytes to interact more easily with the endothelium. This is followed by 4 distinct phases. As seen before, phase 1 begins when activated endothelial cells express P-selectin and E-selectin. This makes it possible for monocytes and neutrophils to interact reversibly through the sulfated thionyl Lewis X expressed on the surface. These interactions form and break rapidly, and this results in the leukocytes slowing down and rolling along the endothelial surface. This step is crucial in order to establish stronger interactions. Neutrophils are particularly effective at rolling on the endothelial surface, even at blood flow rates where other cells are unable to do so. This is thanks to tethers or slings that bind to the endothelium and encircles the cell while it rolls. Phase 2 follows, during which the activated endothelium also expresses high levels of ICAM1 and ICAM2. Moreover, proteoglycans on the surface of the endothelial cells absorb large amounts of chemokine, CXCL8, which binds to the receptor on leukocytes and induces a conformational change in the integrins LFA1 and CR3, thereby mediating a tighter binding with ICAMs, as previously described. This ends the rolling process and results in the adhesion of the leukocyte. When the leukocyte is stationary and firmly adhered to the endothelial surface, phase 3 begins. This is the diapedesis process, namely the actual extravasation. As seen before, diapedesis is mediated by LFA1 and CR3, but also by PCAM or CD31, another molecule of the immunoglobulin family. It is expressed by both the leukocyte and the endothelial cell at intercellular junctions, and it is able to establish homophilic interactions. Once the leukocyte passes through the endothelial cells, it becomes able to produce metalloproteinase MMP9, an enzyme that degrades the basement membrane and allows the leukocyte to migrate into the lamina propria. Once inside the tissue, the leukocyte enters the final phase, phase 4. At this point, the leukocyte follows the gradient of various chemokines such as CXCL8, CCL2, etc., bound to the extracellular matrix. More specifically, CXCL8 attracts neutrophils, which play a crucial role during the first hours of infection, while CCL2 recruits monocytes, which can differentiate macrophages within the tissue. We examined a key immunological process, namely the migration of leukocytes into tissue compartments, hosting an infection or damage. As we will discuss further, this aspect is crucial for the activation of all immune responses, both innate and adaptive.
Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or question, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the following link to visit our website.